Our study object is the Atlantic reef-building coral genus Favia. One of the species, Favia fragum, is distributed along the Caribbean Sea and the Florida Keys, and was also described in the Cape Verde Island. Favia gravida, on the other hand, is distributed along the Brazilian coast and also in mid-Atlantic islands. Despite their allopatric distribution and evidence of genetic differentiation using both mitochondrial and nuclear markers, some researchers don't recognize Favia gravida as a separate species, but as an ecomorph of Favia fragum. The plasticity and the morphological characters traditionally used to distinguish these species, which is a common trait for Scleractinian corals, complicates this taxonomic problem even further. Here we use high-resolution genetic markers obtained through the easy RAD sequencing protocol to assess the molecular support for the current taxonomy. We sample Favia fragum individuals from Panama in the Caribbean and Favia gravida individuals from the entire Brazilian distribution and also one individual from the mid-Atlantic island of Ascension. Our RAD sequencing libraries had very little endosymbiont contamination. And from our assemblies, we recover thousands of single nucleotide polymorphisms and also hundreds of ultraconserved elements, using an Hexacryllia bait set published by Coleman and collaborators in 2020. Both our SNP and UCE trees recover Favia fragum and Favia gravida as highly supported reciprocally monophyletic lineages. Our Brazilian samples were recovered as a highly supported clade within Favia gravida that was separated from the Ascension Island population. These relationships were observed across datasets with different amounts of missing data and matrix completeness. We also observed that datasets with no missing data had the lowest branch support value and appeared to be too strict and failed to recover some of the clades that were highly supported in the remaining datasets. Our results clearly evidence the need to update the IUCN Red List, adding Favia gravida as a separate species with a distribution that does not overlap with Favia fragum. Furthermore, we are able to show that it's possible to recover a significant proportion of ultraconserved element loci from RAD sequencing assemblies using an appropriate bait set. Thank you for your attention, and feel free to contact me in the listed email if you have any suggestions or questions.